2014, Cristo Rey Jesuit was chosen by the Center for Advancement of Science and Space to perform 2 experiments in conjunction with NASA on the International Space Station. We chose 16 students, a team of 8 juniors and 8 seniors. After a period of initial investigation, the juniors decided to study how lipids form the cell wall and form initial life. The seniors decided to study a fascinating creature called the slime mold.
In this project, we learned many skills throughout. Some skills we learned were how to code. We also learned how to program in Arduino, as well as how to solder. And another important skill we learned was teamwork, as we all came together, because some of the juniors and seniors, we didn't know each other as well, but working throughout this project throughout the whole year brought us closer together. And also some of the seniors and juniors planned a major in engineering, which this project benefited us because in the future we'll be able to use engineering basics we learned in this project to whatever college we go to and whatever specific major we choose.
I've learned here in the programming of coding, in the RG lab. I've gone from programming a LED, a simple LED blinking on and off, to programming peristaltic pumps, pumping liquids into the experiment itself.
So during all of our experiments with slime molds, we had to use sterile technique, which is basically to keep everything clean, to make sure that nothing got in the way of the results that we actually wanted to get. So
we worked with techniques such as keeping an open flame to create a bacteria-free environment to make sure that the slime molds were as sterile as possible. We used sterile water, sterile oats, sterile gauze, all sorts of stuff.
Okay, this is all because everything we were doing is microbiology, so since everything is so small, you can't really see what's actually in there that could harm the slime molds. So things such as bacteria or mold that could be in there or could end up growing because of not using sterile technique is what we were preventing.
Yes. Action. Okay, hello, my name is Kelly Wright. I was the junior flyer for the parabolic flight. Flight was amazing. It was really weird and nerve-wracking at first because I didn't know what to expect. But the experience of the zero gravity was weird at first because you felt really heavy. But once it was in the zero gravity part, you just started floating and it was awesome, yeah.
Okay, through this program, I assisted in programming a peristaltic pump, not just one pump, but 5. And we incorporated those 5 peristaltic pumps and 2 Adafruit VC706 cameras, as well as an RTC clock, which is a bit of a hassle. The programming for that was real difficult.
So we started off using the Arduino, which we started playing around with motors and electromagnets, which we saw which ones would react best with our lipids that we 3D printed. We also programmed a camera and a video camera, which allowed us to take pictures and video of what was happening in the reaction chamber, which in the reaction chamber we had to go grind out so we could have a component of 3. In the 3, we set up the camera so they can hone in on what's actually happening. So then we use the vibrations from the electromotor and the electromagnet to see which ones they would get close to in the chamber. We see at what frequency they vibrated at the most.
Okay, so in the past with my job, I've had experience with the CAD program, and so that led us to do our 3D printing designs, which helped us with the lipids and looking at sort of different designs that we could do, and it helped us create the lipids, and we found out that actually the cone lipid was the best 3D printing design that we used. And it's going to help in our experiment later, I guess.
for the advancement of human knowledge and to the greater glory of God, Cristo Rey Jesuit Science.